In this video, we will discuss some important concept of molecular biology. So, in molecular biology, there is a concept called operant concept. So, operant concept is a fundamental concept in molecular biology, and it was first described by Jacob and Monards in 1962. And an operand is a cluster of genes, means it has more than one genes, cluster of genes, and it has what? Promoters regions, operator regions, and the structural genes. The structural genes may be 1, 2, 3, but the main control portions is present in promoter and in operator. So in promoter regions, you can see here RNA polymerase binds to initiate transcriptions. It initiates transcription means mRNA will be generated. If RNA polymerase will bind, that means mRNA will be generated from the DNA as a transcript, and the process is called transcription, and this transcript will be translated to proteins. Then what? Operator, a regulatory region. It's a regulatory regions near the promoter that can either block or allow RNA polymerase to proceed. So it's a control point where the RNA has to pass. So if the regulatory protein or regulatory region is blocked, then RNA polymerase will not proceed and there will be no any transcriptions. Okay, so based upon this, there are two types of operand. One is inducible operand and another is repressible operands. So inducible operand are those operands where the transcription is normally off, switched off, but it can be induced or turned on when a specific molecule, अगर एक specific molecules called an inducer, उसको हम क्या बोलेंगे? Inducer binds to the repressor. See, a repressor is doing what? A repressor is blocking the RNA polymerase to not proceed. Okay, so what the inducer will do? Inducer will bind to the repressor, and by that what happens? The binding side of the repressor is blocked, so RNA polymerase can easily proceed and generate mRNAs. And one more kind of operand is what? Repressible operands. So in this, transcription is normally on, means the process of transcription is going on, but one repressor will come and it will join to the operator region of the gene, and the RNA polymerase will not proceed forward. By that, the transcription will be stopped here. So this operant concept is a basic requirement of an organism. Suppose one organism wants to produce a gene, so what happens? The process of gene transcription will happen. If it does not require any transcription or any translations or process of protein synthesis, then it has to stop the operand. So by this what happens? The energy will be saved. So in this we have understood this operand concept. So suppose this is a genes, this is a gene, and here this region is called promoter, which denotes P, and this region is operator, which denotes O, and this is a genes which needs to be transcribed, and one repressor molecule is there which cannot cannot allow the process of transcriptions because the RNA polymerase, here RNA polymerase will bind, this RNA polymerase will not proceed further. So this is the process, and one inducer molecule is there which will bind to the repressor, and by that what happens, transcription will proceed. This is what, where the gene is in switched off conditions, and in case of repressible gene, repressible operand, what happens, the means gene is continuously transcribed, but the repressor molecule is not bound to the operator. So what happens, the repressor will bind to the operator regions in case of repressible regions, and the transcription process will be stopped. So this is the basic concept of operand. Okay, another thing we need to understand about vector PBR 322. So this vector is used to insert one gene into one bacteria. See, in genetic engineering, what we do, we take the gene of interest, whatever the gene we want to express or we want to multiply into various numbers, we take that gene and insert to a certain portion of a vector. This vector is nothing but one plasmid, which is a DNA, extra chromosomal DNA. In bacteria, what happens, in bacteria, suppose this is a bacteria and there is a gene inside, but one more gene is also present in that bacteria, which is called extra chromosomal gene, and this gene can be used as a vector. So we will discuss one important vector called what, PBR 322. It is a well known plasmid vector. Okay, and it has some key features, like it has some antibiotic resistance gene within the vector. What is that antibiotic resistance gene? That is ampicillin and tetracycline. If this vector is taken up by a bacteria, then that bacteria becomes resistant to the ampicillin and tetracycline antibiotic. So when someone is doing some research, that person can easily grow that bacteria, that recombinant bacteria, because that bacteria has taken up the vector. So that bacteria will grow into that medium containing this antibiotic ampicillin and tetracycline, and that bacteria will easily grow because this bacteria is having the gene that is antibiotic resistant to the ampicillin and tetracycline. By that, the researcher can easily recognize that single bacteria which is a recombinant in nature. Then one more facility is there, multiple cloning sites. See, whenever we want to insert one gene into a vector, we need to cut the gene and we need to insert the gene of interest. Whatever the gene we want to insert into that gene or the plasmid vector, we need to cut that plasmid vector by the help of what? Restriction endonuclease, restriction enzymes. Okay, so with the help of restriction endonucleosome, this enzyme will cut to that vector, and what will happen? By the help of DNA ligase, we insert that gene of interest into the vector. So the vector should have multiple cloning site, means multiple sites which can be attacked by one restriction enzymes. Okay, so that we can cut that plasmid vector, and we can insert that gene of interest. So this PBR322 is having multiple unique restriction recognitions sites. Okay, this MCS allows researchers to insert DNA fragments into the plasmid. So this is the versatile. Then the plasmid size is also very um, good, because it is just 4361 base pairs in length. Okay, so this is good for genetic engineering and replication origins, and it also having one replication origins, means it can be replicated into many genes. Many plasmid can be, means one single vector with the gene can be replicated into multiple vectors with the gene of interest. Then polylinker region is also there. The plasmid also includes a polylinker regions, which is a segment DNA containing multiple restrictions for various enzymes, facilitating the insertions of foreign DNA fragments. So in this polylinker regions, we can insert another genes. Okay, so these all facilities are present in PBR 322. Okay, then one more concept is DNA footprinting. It's a widely used molecular biology technique that allows researchers to study DNA protein interactions, how the DNA and proteins interact with each other, at which points the protein is attaching to the DNA, which particular protein is attaching to which part of the DNA. All these things we can understand by the help of DNA footprinting. So we can understand by the help of this DNA footprinting. Then preparation of DNA. A DNA fragment of interest, typically a few hundred base pairs long, is isolated and labeled. Here labeled means some kinds of isotopes or some fluorescence chemicals will be labeled here, often to one end with a radioactive or fluorescent marker will be used. Okay, by that we can recognize which part is visible. Okay, then protein binding. The labeled DNA fragment is exposed to a specific proteins or a mixtures of proteins under controlled conditions. These proteins can be transcription factors, any protein, DNA binding proteins, or other molecules that interact with DNA. Then what happens after the labeling of the DNA? What happens? Protein binding will be happens. Okay, then chemical treatment will happen. This here what happens, the chemicals which can clips the DNA at specific sites will be used. This DNA, these chemicals are nothing but the nucleases enzymes which clips at specific sites. After that what happens, the clipped DNA, those DNAs which are broken down into small pieces, are sent into or treated with what, are separated by using what, gel electrophoresis, where the shorter fragments migrating further through the gel. Means this gel electrophoresis is nothing but the separations of the DNA molecules based upon the size. So the small molecules of the DNA will move faster. Then what happens? Visualizations will happen by the help of uh, X-ray films or radioactive levels scanned or for fluorescence. The result is one radiogram will come. So auto radiogram will be seen like what? Auto radiogram will be seen like this. Suppose this is the gel. So here like this separations. This is the auto radiogram, where this auto radiogram will be compared with one controlled DNA, and by that we can see that which portions of the original DNA is interacting with the protein. So by this we can understand that interactions between proteins and the DNA, or we can understand how the proteins is attaching to which point, and different biological processes we can understand by this process. Okay, then next is DNA fingerprinting, also known as DNA profiling or genetic fingerprinting. So this is used in forensic technique, where we can use it's in forensic technique. We can use it for identifying the individuals based on their unique DNA fragments. See, unique DNA pattern. See, all individuals is having a different DNA patterns. So we can recognize that DNA pattern by this process of DNA fingerprinting. So by identifying that particular pattern, we can recognize that this profile or this particular pattern is belong to this particular persons. Okay, so first we have to do what? Sample collections. We have to collect the sample by different method like swab analysis, blood samples. Everywhere, anywhere you can take the samples, different fluid of the body you can take, and because that portion is having DNA, and this particular technique is utilized in forensic science, where suppose upon some crime scene is happened, and in that crime some bodily fluid is found from the accused, and that bodily fluid will be taken as sample, and whoever the person is in doubt, that person's fluid will be taken, and the DNA will be isolated, and the particular pattern will be matched in between these two sample. Then what we do? The DNA extraction will happen. Different techniques is there for DNA extractions. Then PCR. What is PCR? PCR is nothing but increasing the patterns, means increasing the desired site of a DNA. See, all the DNA is having some patterns. So what we will do, we will recognize the particular patterns, okay, which is also called as small tandem repeats, and we will amplify, that is called amplifications. We will amplify that particular STRs or small tandem repeats, and we will do what? We will recognize that patterns. These STR regions are also known as loci. Then what we have done after the amplifications of that STR, small tandem repeat, amplification means nothing but increasing the number of that small tandem repeats. Small tandem repeats means repetitions of the base pairs. This is called small tandem repeats. After what, we have to amplify or increase the number. After that, that increased quantity of that patterns will be gel electrophoresis. Okay. Then what analysis will be done? Analysis, the pattern of the DNA fragment is analyzed. The number of repeats at each STR locus and their sizes are determined. These informations constitute the individual's DNA profile. So one profile is generated, and after that what we have to do? We have to do comparison. Then DNA profile from different samples such as crime scenes, evidence, or any other points are compared with that patterns. Okay, this DNA fingerprinting is highly useful during the investigations of any crimes, and it is fully approved. Next is, what is Cosmet Vector? See, when we want to clone or insert one big DNA fragment, we need Cosmet vectors in molecular biology or in genetic engineering. So this Cosmet vectors combines features of plasmids of bacteria and bacteriophage. Bacteriophage is nothing but the virus that infect a bacteria. So a bacteriophage or a virus will give one part that is called cos part and one part of bacteria that is plasmid, and by attaching these two we will get what? Cosmet. Okay, we will get what? Cosmet. So suppose we have one example here. See, this is nothing but a what? This is a bacteria. Okay, this bacteria is infected by this virus. This is called lambda virus, and it is having one DNA which is called cos site. And what happens? This gene will enter into this bacteria and it will attach with this genes. And what happens? When the bacteria will multiply, the cos site or the genes of virus will also multiply into 1000 numbers. We will utilize this phenomena and we will do what? We will generate one library, gene library or DNA library. Okay, so this Cosmet vector is having what? Two parts, one from cos side and another from plasmid side. So that's why it's called Cosmet vectors. Okay, so this Cosmet vector is having what? Origin of replications, which helps in multiplications, I mean start of replications. Then cloning sites, where we can recognize the breakdowns or not breakdowns. We will make a nick in the DNA segments. Then cos sequence, already discussed. Cos sequence is from what? The virus. Then selectable marker. By this we can find out whether the recombinant genes is living or growing or not. Okay, we can recognize this, already we have discussed. So selectable markers are nothing but what? Resistance, antibiotic resistance. Okay, then we have already told this large DNA insert capacity is what? It has the capacity to insert 35 to 45 kilo base pairs of genes or DNA. Then it can also help in development of library, cosmic library constructions, where we can use DNA fragments. We can use what? Big DNA fragments and make a collections of entire genome of an organisms. Entire genome of an organisms can be collected and made a libraries. These libraries are very valuable for different studies. Okay, then what about a typical vector? We have discussed PBR 322, plasmid vector. So what should be a typical vectors to use it for different purpose like cloning, in genetic engineering, everything. So it should have an origin of replications. So by that, the DNA sequence that allows it to replicate automatically with the host organisms. So it should have one origin of replications that allows it to replicate in the host organisms automatically. Selectable markers, some antibiotic resistance should be there in that vector. These markers allows to the identifications. Then cloning site, it should have MCS or multiple cloning sites. By that we can insert our desired genes in that cloning site, because these multiple cloning sites can be clipped by the help of restriction endonuclease, and we can insert our desired gene into it. If this cloning site will be not there in the vector, then this is not called a vector. Okay, and we cannot insert the gene, so it is must. Then promoter and terminator sequence. If the vector is intended for gene expressions, agar if you are going for expressing the genes, it needs to be having promoters or terminators. The promoter will initiate the transcriptions of an inserted genes, while the terminator signals the end of transcription. So it's a regulatory area, means this regulatory promoters and terminators should be there if it is an expression vectors. Then minimal sequence, to minimize the interference with the functions of the inserted DNA. See, our main purpose is the inserted DNA, desired interested genes. Okay, so the vector should not interfere with the actions of the inserted genes. So the vector should have minimal DNA sequences, so that there will be less interactions between the vectors genes and the genes of interest. Then stability and copy number control. Depending on the application, vector may need mechanism to control their copy number within the host cells. See, the vector should have less copy numbers if it is an expression vector, and its stability should be, it should be stable inside the host cells. So this is a good characteristics to be typical vectors. Then size, vector should be a manageable size, we have already discussed. Then compatibility with the host organism, it should compatible with the host organisms, so that it can be inserted into the host organisms and it can be stable within it. Then sequencing and mapping, vectors may include sequencing primer sites and known DNA sequence that make it easier to sequence and map the inserted DNA. Then specialist features, depending on different experiments or applications, vectors may have additional features. Suppose some vectors will have some coloring, means some reactions between some molecules will generate the color. All these extra features may be there. So the choice of vectors depends on the specific needs of the molecular biology experiment. So with this we have understood some important concepts of molecular biology, which are very helpful for our genetic engineering. Thank you.